Every known organism on Earth follows a strict unidirectional sequence. Life progresses from birth to growth, on to senescence, and finally, inevitable death. For decades, biological aging has been viewed as an inescapable law of thermodynamics. But hovering in the Mediterranean Sea is a biological anomaly that breaks this rule. This is Turritopsis dohrnii, commonly known as the immortal jellyfish. The organism is small enough to fit on a human fingertip, measuring no more than 4.5 millimeters across. When confronted with the natural end of its life cycle, it possesses the physical ability to hit a biological reset button. Its existence proves that the presumed permanence of cellular differentiation is actually reversible. The eminent German zoologist August Weismann first described the species in 1883. For over a century, scientists categorized it as an ordinary hydrozoan, assuming it lived, reproduced, and died in the exact same pattern as its relatives. That assumption held until 1988, when a marine biology student named Christian Sommer was collecting Arius hydrozoans in Rapallo, Italy, keeping them in petri dishes to study their life cycles. Instead of watching his adult specimens age and die, Sommer observed them seemingly dissolve. The adult medusae morphed directly backward into their sexually immature, juvenile polyp stage. He had inadvertently documented reverse development. Under specific laboratory conditions, the organism's biological clock could run backward indefinitely. The reversal mechanism does not happen at random. It is triggered by extreme environmental stress, a sudden drop in temperature, extended periods of starvation, or severe physical injury. Once stressed, the jellyfish enters a phase of morphological deterioration. It ceases swimming, its gelatinous bell shrinks, and it actively resorbs its own tentacles. Within 72 hours, the remnants of the medusa sink to the ocean floor and condense into an unorganized, spherical mass of tissue known as a cyst. While this outward collapse resembles decay, the cyst is actually a highly protected, dormant environment. It acts as an armored shell where a complete biological teardown and rebuild occurs. The mechanism driving this transformation is transdifferentiation. Instead of dying, fully mature specialized cells revert back to an unspecialized stem cell state. Inside the cyst, the organism undergoes massive cellular apoptosis. Anatomical structures no longer needed undergo programmed cell death. The surviving cells strip away their epigenetic markers. The molecular tags that locked these cells into their specific adult identities are completely erased. Rather than simply healing a wound, the jellyfish dismantles its highly specialized anatomy and reprograms its existing cells to construct a younger version of its own body. In 2022, researchers at the University of Oviedo published a comprehensive genomic sequencing of the jellyfish in the Proceedings of the National Academy of Sciences, mapping its genome to isolate statistical anomalies. This table compares the immortal jellyfish against a mortal relative. The top row reveals a massive expansion of genes for DNA repair. Further down, mutations in the POT1 gene complex increase telomerase activity, preventing chromosome ends from shortening during cell division. Researchers also hypothesize the jellyfish actively targets a protein complex called PRC2, which normally locks cells into their specialized states. During stress, the organism bypasses this silencer, unlocking the DNA to allow cells to change their identity. After days of genetic reprogramming, the dormant cyst begins to sprout root-like stolons, anchoring to a rock and growing into a new polyp colony. The organism's genes execute a precise set of instructions to birth hundreds of genetically identical clones directly from the remnants of a single adult medusa. This complete physical overhaul presents a biological version of the Ship of Theseus paradox. If it dismantles its neural net to build a clone, whether it remains the same continuous individual is an open philosophical question. The jellyfish is also not completely invincible. It frequently succumbs to extrinsic mortality, meaning it can be eaten by predators or killed by a rapid viral load before it has a chance to form a cyst. Yet, by relying on transdifferentiation, the organism has functionally erased its intrinsic mortality, the internal degradation resulting from biological age. If scientists can isolate the exact proteins that trigger this cellular transdifferentiation, it offers a distinct model for human regenerative medicine. Therapies could theoretically coax mature, damaged human nerve cells to revert to a stem-like state and regrow connections after severe spinal cord injuries. Because this reprogramming requires strict regulatory control over rapid cell division, studying how the jellyfish safely executes this process without forming tumors could also yield new targets for anti-cancer therapies. The ability to revert cell identity without losing regulatory control provides a biological framework for repairing physiological damage that was once considered permanent. The jellyfish's genomic blueprint for advanced telomere maintenance and DNA repair provides a concrete roadmap for future pharmacological interventions in humans. Genomic data shows the jellyfish maintains a superior defense against reactive oxygen species, the volatile molecules that drive cellular aging. By mimicking these specific pathways, scientists hope to slow the oxidative damage that contributes to the progression of Alzheimer's and Parkinson's. Turritopsis dohrnii stands as a reminder that biological decay is not a universal constant. It is a process that can be halted, reversed, and repeated.